Hi, my name is Zoe Reilly, and I'm going to present my journal club review for this article, Diet Outperforms Microbial Transplant to Drive Microbiome Recovery in Mice. I picked this article because I have a deep interest in human gut health, especially since I have been on my own journey with this topic. I also picked this article because any way out there to optimize microbiota recovery following antibiotic treatment has my attention. Probably the biggest reason, though, for me choosing this article for review was due to researchers' observation of two different diet types that interest me, one of them being the Western diet. Since I live in the US, the Western diet definitely affects my environment, and honestly, in the past, I have found comfort in this way of eating from the self-nurturing aspect. So while simultaneously wondering how good or bad this diet is while I'm eating it and what it has, how it's affecting my gut has definitely been on my mind. So why care? A healthy gut is incredibly important for our overall health. Speaking for humans, 70% of our immune system is stationed in our gut. The amount of nerve endings in our gut has a large portion of the academic community referring to our gut as our second brain. Gut disorders like intestinal bowel syndrome are being found to be linked to mental disorders like anxiety and depression. And overall, the gut is um, it's just important. It's a major pit stop in how we gain our energy, our fuel, and our nutrients. Our diet, as the host, has a direct correlation with our microbiome metabolic activity. Let's look at the facts. We want a diverse community of microorganisms in our gut microbiota, and a diverse community of microorganisms would likely require a diverse diet, as differing microorganisms require differing nutrients to survive, proliferate, and colonize. So as I stated before, this article focuses on two different types, our diet types, one being the Western diet. I feel like as um, I feel like as we plan to continue and increase our industrialized presence, our food source has definitely become more industrialized. The focus is not on being industrialized for this discussion. It is the nutrient content associated, that being low fiber and high fat. Is this the way to a healthy and happy gut microbiota? Stay tuned. So questions being asked. I would consider there to be one primary question for this article and one secondary question that were asked that drove the basis of this research. The primary question is, how do dietary conditions influence recovery of the gut microbiome after antibiotic treatment? The secondary question is, how does this dietary influence compare against the effectiveness of microbial transplantation? Okay, so was the research goal achieved? Yes. Yes, it definitely was. The research demonstrated the importance of fiber in the diet and the association between the Western diet and severe and prolonged microbial gut dysbiosis following antibiotic treatment. The research also demonstrated that complex carbohydrates promote development of complex communities in syntrophy. This was demonstrated through in vivo ecosystem creation through the utilization of mice. The research also achieved observation of prolonged or profound redox imbalance within the gut that was associated with the absence of fiber following antibiotic treatment. I'll go more into detail about this later, but this redox imbalance definitely aided dysbiosis. Okay, so far, how far has the gut microbiome research come to make this point, aka the current research article? If my research, in my research, If my research of documented timestamps serves me correctly, Antoni van Leeuwenhoek seems to be one of the first documented gut microbiome researches. In 1681, Antoni examined our watery stool under a microscope and observed, quote unquote, more than 1,000 living animalcules. So 300 years in the future, Nature magazine would publish an article discussing gut microbiome differences observed across 531 individuals amongst various populations. This article would also detail the impact of westernized society versus not in association to the observed individuals. In 2021, a study conducted in Spain observing diet association with the human gut microbiome was published. This study observed 530 healthy volunteers and revealed similar microbiome patterns amongst individuals who ate meals that fall within the Mediterranean diet. Okay, so what is the urgency for this subject research? Generally, in current state, antibiotics tend to be the first line of defense in regards to bacterial illness. Antibiotics are common in industrialized populations, with most people listening to this journal review most likely having taken an antibiotic in some form in the past year. The aspect of the Western diet concerning high fat, low fiber is proven to be very prevalent in the American population. A journal published in 2023 stated that 72% of daily calories consumed by the US population is made up of dairy products, cereal, refined sugars, and oils, and alcohol. Most, if not all, falling within that high fat, low fiber bracket. Why not continue to reflect on the pros and cons of any diet, let alone a diet in a country that is not shy to disease? Okay, so let's dive into this article. And to note, the rest of these slides and information I'll be pulling are really directly from the research paper. Although previously mentioned, I wanted to list this in a little more detail. The research team to set out an answer to how dietary conditions influence recovery of the gut microbiome in mice following antibiotic treatment. Specifically, the research team observed how the Western diet and quote-unquote regular chow diet compared in influencing gut microbiome recovery following antibiotic treatment. The low fiber, high fat diet was titled the Western diet. The low fiber, let me start that over again. The low fiber, high fat diet was titled the Western diet. The high fiber, low fat diet was titled the regular chow diet. The research team then observed how these individual dietary influences compared against the microbial transplantation, specifically fecal matter transplantation, or FMT. Okay, so their experimental overview. This figure provides a pretty good visual for the experiment's procedure. 86 female specific pathogen-free mice were acclimated to either the regular chow diet or the Western diet for 4 weeks following initial microbiome homogenization in their new environments, aka cages. After 4 weeks, the mice from both dietary groups were treated with either a triple antibiotic cocktail liquid treatment or a 5% phosphate buffer saline liquid treatment over a 72-hour time frame. The mice remained on their assigned diet throughout their 3-day liquid treatment. Following their liquid treatment, the mice groups recovered for 4 weeks, remaining on their assigned diets. You can see where the fecal samples were collected, which was throughout the study and most frequently post-liquid treatment. There was a long-term cohort who was sampled for 9 weeks versus kind of the standard 4 weeks for both groups following consumption of both liquid treatments. And there will be some data that coincides with the 9 weeks. Of note, there was one additional cohort of 12 male mice that underwent the same protocol as the female mice for up to 2 weeks post-liquid treatment consumption, and that was to confirm the recovery patterns were consistent for both sexes. Because the results showed that recovery patterns were consistent across both sexes, only the analyses that are included for female mice are reported unless specifically stated. But just know that that was confirmed, so it's not just a female. Okay, so to ensure successful understanding for future images and discussions, I just wanted to clarify some abbreviations and terminologies that will be employed. I will also share some methods utilized for the various analyses. So RC stands for regular chow. This is the high fiber, low fat diet. WT stands for the Western diet, which is the low fiber, high fat diet. ABX stands for antibiotics. PDS stands for phosphate buffer saline. ASV stands for amplicon sequence variant. Gene calls are essentially an open platform profiling technique of mRNA transcription. Gene calls measure gene expression levels of specific transcripts. Orthologs are genes that evolved from an origin gene in a species and are now present in multiple species through speciation. KGG is an ortholog database, and just a reminder that syntrophic interactions are cooperative interactions between at least two microorganisms to downgrade a substance that can't be metabolized by that one species alone. Okay, let's discuss some of the methods used to perform this analysis. So microbial biomass was determined via colony counting. Colony counting was executed via plate counting or counting of colony forming units or CFUs on culture-rich media plates. The gut microbiome taxonomic recovery was determined via 16S RNA gene sequencing. The sequencing was executed via Illumina sequencing, and data was analyzed with QIIME2. Microbiome functional capacity pre and post antibiotic treatment was assessed by shotgun metagenomic sequencing of fecal samples. Metagenomic sequencing was executed and assessed using the Illumina NetSeq 500. Gene calls were annotated utilizing the Kyoto Encyclopedia Genes and Genomes, which is the KEGG, KEGG catalog for functional interpretation. And statistical methods were organized per experiment and will be shown in future figures. Okay, research findings, observations, and results. Okay, so microbiome biomass recovery. So figure 1b illustrates, which is on the screen, illustrates each diet group's microbiome cell counts, pre-antibiotic treatment, during antibiotic treatment, and post-antibiotic treatment. For both diet groups, microbial biomass counts decreased rapidly following antibiotic treatment, with counts of the Western diet mice declining much more severely than counts associated with the regular chow diet. At day 4 post-antibiotic treatment, the regular chow diet colony-forming units returned to baseline from prior to antibiotic treatment, and that was for both anaerobic and aerobic organisms, whereas it took till at least day 7, more so day 8 for the Western diet biomass numbers to return for just aerobic baseline bacteria, and then closer to day 13 for the anaerobic bacterial counts for the Western diet. And then the article states that the PBS control mice for both diets remain stable across both treatments. Diverse taxonomic recovery measured via Faith's phylogenetic diversity showed a sudden decline in both groups upon antibiotic treatment. Following antibiotic treatment, regular chow diet mice recovered more than half of their initial diversity by day 11. However, the Western diet group observed severely diminished phylogenetic diversity for up to the measured 28 days in all cohorts. And even for that 9-week longer cohort, it was pretty diminished. You can see that it's really not increasing there. Phylogenetic diversity with the PBS control groups did not change significantly, as you can see. Okay, so relative abundances of taxa within the gut were significantly altered for both diet groups during and after antibiotic treatment, depicted here in figure 1D. In the regular chow antibiotic group, their microbiota experienced sequential stages of recolonization marked by early dominance of facultative anaerobes such as Enterococci, Lactobacillus acei, and Erysiphalatrixaceae, which was followed by increased diversification of stricter anaerobes. In the Western diet antibiotic group, the low biomass post-antibiotic consumption gut flora community was dominated by Streptococci, a facultative anaerobe, until microbial biomass numbers started to recover. Around day 14 post-antibiotic treatment, the Western diet antibiotic group also passed through a sequential stage of facultative aerobic colonization, which included similar key families as the regular chow group. before recovery of stricter anaerobes. Something I do want to note is that I think it's super cool the intense taxa abundance change witnessed at day 0 of antibiotic treatment. I love the visual of it, especially comparing the two groups with the regular chow on the top and the Western diet below. By day 63, which is essentially 2 months post antibiotic treatment, you can see how both groups don't have like for like taxa abundances as prior to the antibiotic treatment. However, you can see that both groups do show similar families returning at day 63 that were present in each group prior to the antibiotic treatment, which much more diversity observed in the regular chow diet group. Okay, so functional richness. Microbiome functional capacity was assessed through performance of shotgun metagenomic sequencing on fecal samples taken from subsets of mice from both diet plus antibiotic groups. Key time points including pre-antibiotic treatment throughout recovery. So looking at the extended data figure 2A on the screen, gut microbiome functional diversity was determined by calculating functional richness at various hierarchical levels. At the level of gene calls pre-antibiotic treatment, mice in the Western diet experienced reduced functional richness compared to the mice on the regular chow diet. Post-antibiotic treatment, functional richness plummeted significantly for both diet groups, not surprising. However, where mice in the regular chow diet recovered up to 69% of their initial gene count by day 28, mice on the Western diet on day 28 only recovered up to 16% of their initial gene count. At a broader hierarchical level, such as the KEGG ortholog, abbreviated via KO on the screen, or the KEG category, abbreviated KCAT on the screen, functional richness was preserved during and after antibiotic treatment for both treatment groups. These results can indicate a change in the number of unique gene calls that map each of the KEG orthologs before and after antibiotic treatment, potentially indicating a change in functional redundancy. When this was assessed, further, it was observed that mice in the Western diet experienced a loss in functional diversity and redundancy following antibiotic treatment that varied in intensity across KEG ortholog groups, with notably major losses in metabolic redundancy. Okay, so functional characteristics of each microbiome group was compared in a pairwise manner across all selected experiment time points. Post-antibiotic treatment depicted in both figures on the screen measured regular chow mice to have 658 significantly depleted KEG orthologs at day 2 post-treatment, post-treatment being post-antibiotic treatment, compared to the pre-antibiotic state. Interestingly, this number grew to 835 depleted KEG orthologs at day 4. I found this interesting as the graph. I just found it interesting that it would increase a little bit and then decrease. I just didn't expect that. I would consider that most of the depleted orthologs would be as close to the antibiotic statement or treatment as possible. However, for the regular chow group, nearly all KEG orthologs recovered to baseline levels by day 14, which unfortunately mice in the Western diet had a large number of significantly depleted KEG orthologs compared to the regular chow numbers at all time points, even by day 28. So you can see that the depleted numbers for the KEG orthologs were pretty much gone by day 14 for the regular chow, whereas the Western diet, even by day 28, you had still notable gene depletion. Okay, so here's a heat map of available resources within the gut environments for both diets. So functional recovery was also examined throughout the assessment of available resources within the gut's environment for each group. This was conducted through targeted fecal metabolic compounds, including amino acids, carbohydrates, bile acids, fatty acids, and others for both mice groups pre and post antibiotic treatment. It was observed that mice in the regular chow diet saw a distinct drop in normalized abundances of many compounds from baseline after antibiotic treatment, and that the compound abundances had returned to baseline numbers by day 11. This was statistically confirmed by PCOA clustering analysis, which is extended in figure 3C. In extreme contrast, the metabolomic profile of the Western diet shows that there was almost no signs of recovery until minimally day 14. Personally, for me, I actually find this heat map graphic slightly hard to use for visualization purposes. However, I did choose to show this so that you could observe one aspect of the heat map that is very easy to observe, and that is your bile acids, which are heavily depleted throughout sampling for the Western diet. So bile acids depleted for the regular chow diet, day 0 to day 7, were then in the positive, whereas you can see for the Western diet, it was just constantly in the negative. Okay, for the next 3 slides, measures of available short-chain fatty acids were measured and documented separately. Regarding regular chow mice treatment group, concentrations of acetate, butyrate, and propionate were statistically indistinguishable for those of pre-antibiotic measuring of at day 14 and day 28. That's for the regular chow. Whereas the Western diet mice treatment group, the same subject short-chain fatty acids were depleted at day 14 and persisted at low levels to day 28. The data across these 3 figures is significant as short-chain fatty acids are well-documented products of microbial metabolism and directly affect the host. So here's the graph for acetate. Here's your graph for butyrate, which you can just see pretty low level. And same with propionate. Okay, so of all the figures, this is my favorite illustration. Across all taxonomic and functional metrics that were evaluated, mice in the low-fiber, high-fat Western diet experienced a more severe ecosystem collapse and a slower and less complete ecosystem recovery than mice in the high-fiber, low-fat regular chow diet. It was noted within the article that this severe difference was not linked to slower antibiotic clearance between either group. Figure 3A shown on screen is an illustration created from community flux simulations conducted over each time interview for antibiotic-treated mice of both diets. When looking at this figure, you can clearly see the difference in metabolic activity and key players between the regular chow diet, which is on the top, and the Western chow or the Western diet mice across negative day 3, which was the first day of antibiotic treatment, and day 14. Note how the Western diet mice are dominated by Lactococcus amplicans sequence variants, day 0 to day 7, whereas you can see more community metabolic activity with the regular chow mice. And on this next slide, you can note the difference in syntrophic interactions across both diet treatment groups. I also really love this illustration as well. You can see that there was a decrease between both groups with the introduction of antibiotics and actually complete absence of interaction at one point for the Western diet treatment group. I also think it's important to note how even in the beginning of syntrophic interactions for regular chow were greater in quantity and to me depicting a more supportive and stronger microbiome community even prior to antibiotic treatment. Okay, so let's pivot into the part of the article involving dietary intervention influence compared to microbial transplantation influence via fecal matter transplant. The research team believed that the limited microbiome recovery observed in Western diet mice was associated with the imbalance of availability of simple and complex carbohydrates rather than lack of metabolically capable taxa. With this hypothesis, the researchers performed an intervention experiment to test their theory, where diet and microbial re-exposures were controlled after antibiotic treatment. In short, after antibiotic treatment, mice from each pre-antibiotic diet were transferred into sterile gnotobiotic cages, which are highly controlled environments for germ-free or defined microbial flora studies. The mice were fed their respective post-antibiotic treatment diets and then experienced microbial re-exposure via fecal matter transplant administration. FMT, which is the fecal matter transplant abbreviation, was given to the mice 24 hours post antibiotic treatment and again at day 14. Day 14 was as well selected as the time to evaluate distinct recovery dynamics in fecal samples. I will note I do not understand why the researchers chose day 14 for the second fecal matter transplant. If someone thinks they understand why, please feel free to comment and give your thoughts during the journal club discussion. However, as you can see from the figure on the screen, diet clearly had a notable effect on ASV richness and recovery, which I found cool to see. To explain what you're seeing, the box plots on the left are the experiment or the experiment's PBS treatment controls. So that is considered our baseline. In the middle and right side of the figure, we have comparison of regular chow versus Western chow pre-antibiotic diet mice who were given either the same or a different diet post-antibiotic treatment. This post-antibiotic diet differentiation is labeled with that D subscript, and the M subscript represents the microbial transplant diet organisms given via fecal matter transplant. At the bottom row, you'll notice that in some cases PBS was given in replacement of regular chow or Western diet organism fecal matter transplant to serve as a control. Among mice that were fed post-antibiotic Western diets, the right side of the graph, you can see that FMT showed very little impact on microbiome activity recovered, and all Western diet treatment groups exhibited extreme ASV richness diminishment. However, you do not see this lack of richness with the post-antibiotic regular chow diet groups, even amongst the mice that were fed the Western diet prior to antibiotic treatment. The article's authors stated that this experiment, illustrated on the screen, indicated that an appropriate diet is both necessary and sufficient for rapid and robust gut microbiota recovery post-antibiotic treatment. Personally, it took me a while to see the graph in the same way as authors for some reason, but after some time, I finally understood and agreed, especially when remembering this middle section is the recovery post-antibiotic treatment, whereas the left-hand side never saw antibiotic treatment. And just for those who are interested or this may help, this following figure illustrates the methodology of FMT influence versus diet influence experiment in a more visually and clear way. Okay, to circle back to what we had introduced at the beginning of this presentation, I think it's much more clear now that a healthy gut microbiome, aka diverse and metabolically active gut microbiome, is incredibly important for overall host health. And what we learned through this paper is how we gain this healthy, diverse, and metabolically active gut microbiota is through a diet high in fiber and low in fat. Through this research, we saw that the Western diet has an unhealthy crippling effect on our gut microbiota, especially and specifically during and following antibiotic treatment. With the Western diet continuing to grow its presence globally, this does not seem like the correct path for society for healthy gut microbiota. Or at least I think that medical practitioners, if they're going to be giving antibiotics, it's almost a responsibility for, in my opinion, for them to be clarifying a successful diet for their patients to be having, aka high fiber, low fat, during and following antibiotic treatment. What I'm curious to know, and what we did not find out through this paper, is dietary influence on gut microbiome health and recovery if the diet was both high in fiber and high in fat, and what host diseases may result due to the lack of microbiota metabolic activity in the gut if consuming the Western diet during and post-antibiotic treatment. So conclusion, the Western low fiber, high fat diet showed severe ecosystem collapse during and post-antibiotic treatment compared to the high fiber, low fat diet. The high fiber, low fat diet showed impressive microbiome recovery post-antibiotic treatment across all experiments, including recovery of biomass, taxa diversity and abundance, metabolic activity, and syntropic interaction. And diet compared to FMT has a significantly larger influence and effect on microbiome recovery post-antibiotic treatment. Okay, so I came up with some 5 thoughtful questions for the discussion. These questions include, number 1, did you expect to see such significant differences between the 2 diet types in regards to microbiome recovery? Question 2, What did you appreciate the most in regards to the data presented? Question 3, what do you wish you learned more about in regards to this research? Question 4, has this research changed how you may approach diet in the future, especially in regards to diet during and after antibiotic treatment? And question 5, what specific diet types would you like to be researched next in regards to microbiome recovery and why? I really hope you enjoyed this research article presentation and I'm looking forward to our back and forth discussions. Thank you.